Hello everyone. Hello everyone. And yes, I can hear myself and welcome again to another Saturday microscopy live stream. I'm Oliver, Microbehunter. That's how I call myself. Yeah, just give me again a couple of seconds to get everything sorted out again. I think sound seems to be working because a few months ago, I even I lost my sound and yeah. I think everything's fine. Okay, so hello around the world. Today again, a little bit of more or less random microscopy. You see and hear me loud and clear. Thank you very much. Hello all around the world. And yes, all the way to the very first comment, my inbox was full. Now it's not full anymore. So if you want to, you should be able to send me emails again. Hello to North Carolina, to Oregon. Atlanta. All over the place. Athens, Greece, Vietnam, Netherlands, New Jersey, US, Florida, Upper Austria, Netherlands, okay, London, Washington. Yeah, it's really nice to have such a nice, nice international community here. I see that some of you have already exchanged call signs because you're ham radio operators. Several years ago, I was also considering going into that direction and to pass the amateur radio exam. Decided then not to, but in any case, nice that you were able to join again. So very briefly today, every time when I prepare something for the live stream, I'm kind of thinking a little bit of something new. Sooner or later, the things repeat a little bit. And today, I just want to show you also something that I tried out a couple of some time ago. And these are the cells of a banana. And what you see in there, these are starch grains. I want to show you now a very brief preparation technique, and then later on we can do a little bit of staining as well, especially with iodine, for example. So these are starch grains. Of course, potato, a very common one. I've done this already many times before, but let's have a look at bananas now. Then later on, of course, I also want to have a look at the sample, the pond water sample where I made an enrichment culture, whether the ciliates, paramecia, and so on, whether they actually started to grow already or not. We're going to see. Yeah, and I have a couple of other random things, and we're going to just going to take it from there, and we're going to see how far we can get. If you have anything specific, anything specific that you want to see, please inform me. Okay, just a second. Okay, so what I'm going to do is the following. First of all, I've got so many people joining in, some more experienced, some less experienced, and very often I'm getting the question, okay, how do you get those colors, right? I just want to show you, say it again, I've got special optics that give a little bit colors, right? I can even change the colors around, but in regular bright field, it will look like this. I know it looks a little bit less exciting, but just to tell you, if you want to also observe banana cells, that's probably how you're going to be able to see it. Okay, so that is just something I just wanted to make clear here. So what I'm going to do is the following: is I'm going to first make show you the traditional way, and the traditional way which does not always work very well. Okay, and then I'm going to show you the other way, and we're just going to get started, and you can basically just talk about all sorts of different things as we go along. Okay. Are bananas suitable nutrients for ciliates and other critters? I say, why not? Because they are very, not very specific. They eat pretty much everything. And ultimately, I have to tell you the following. Even if you add starch grains or bananas, it doesn't matter. Because those ciliates, what they do is they do not feed directly on the banana cells. The banana cells and the starch is food for bacteria. So bacteria are going to start growing. And then the ciliates, they feed on the bacteria. So what you're doing is you're creating a small food chain. Yeah? So that's always very important. When we make enrichment cultures, the ciliates generally don't feed on the actual food that you give it. It's still too big, right? But bacteria feed on those. So I'm going to show you first so this banana, I've just cut it just a few hours ago. I'm going to show you now the traditional method, so to say, and you take a sample here. You put a sample on a microscope slide like this, okay? Way too thick, of course. Okay, and then what you do is you add a cover glass. Yeah, here it is on top, and then you got to be really careful that you don't damage the slide. You add a tissue paper on top. Okay, and then hopefully without breaking the cover glass and or slide, you press down vertically. No horizontal movement. Okay, no shear forces. This is called a compression specimen, and you see that it's a very thin, some of the banana squeezed out here, but generally you get a very thin film. Look, even the cover glass, I was sliding it horizontally. Yeah, we're just going to have a look at this first. I'm going to show you the advantages and disadvantages of this method. Okay, that is how do you say, a very common technique with soft tissue. If you also want to prepare, for example, not onion root cells, you do the same thing. Yeah, and I'm just going to show this to you now. I have to remove the other slide. And at the same time, I always have to switch also, yeah. We're gonna see all of the advantages and disadvantages of this method, yeah. That's basically how it looks like. Actually, it looks better than expected. Okay. Actually, I have to tell you right now, I just realized the following. This worked better than expected, so that the other method actually is totally obsolete. No, I'm going to show you the other method in any case. So what we have over here is, of course, the individual banana cells, and those round circles that you see here, air bubbles, right? Yeah. What I'm going to do also, I'm sometimes also going to look at the whole thing a little bit at a slightly lower magnification. Okay. And then occasionally I'm also able to see some of the xylem vessels of the banana. Okay, and but not over here. Yeah. So yes, how do they look like under cross polarized light? That's also something that I was kind of expecting that some of you wanted to see. So what I do have polarization, right? However, I also have other prisms that change the color. But if you actually want to observe them in regular conventional cross polarized light using some just regular polarization filters, these are linear polarization filters. I'm also going to show that to you. Okay, so I'm going to remove the polarization from here. Yeah, I'm going to remove the polarization from the top. Yeah, so it's now completely like a regular bright field. And now I have to add a filter on the bottom and at the top. Okay, so this is going to be super improvised now. Okay, no, I'm going to put do it like this. I'm going to add one at the bottom. Yeah, so over the lamp, and the other one I'm going to add between the cover glass slide and the objective. Okay, so I have to go up with of course the focus changes because of that. So I have to refocus, and now I'm going to rotate one of the filters, the one over the lamp, and then it will look like this. Okay. Yeah, so you see that they are a little bit, The problem is I cannot go up with the magnification right now. So how am I going to do this? I have my other filters somewhere. I'm going to change those. So I'm going to put the thinner one over here. Okay. So that is now using, again, cross-polarized. Let's see if I'm able to go up a little bit further. No. I think I can't. Or can I? Yes, just barely. Okay. Yeah, here we go. So that is basically now, just in standard regular cross-polarized, cross-polarized light. Okay, it's a little bit risky because it's I'm almost touching the filter, but it's going to be fine. Yeah. So basically, as you've mentioned, the following, yeah, ideally you would, let's try this, you would actually put the filter directly beneath the slide because you've got all of the condenser optics and everything in between, which also might depolarize everything. So I'm going to do the following is I'm going to take this, you have to now watch a little bit what I'm doing up there, right? I'm going to now take this out. I'm going to put a filter directly over the condenser like this. Then on top goes the slide, and then yeah, I have to add, and I have to check which orientation is the right one, and let's do it like this, and I have to now kind of find it. Here we go. Okay, and I'm going to have to rotate this to make it darker. Maybe not too dark. Let's go up a little bit here. And we can refocus, and then maybe we're getting a slightly better image quality because we've basically eliminated the condenser. Okay. Yeah, so that's yeah, it's a little bit, maybe a little bit too much, but you get the idea. So there's plenty of opportunities, yeah, to play around here. Yeah. So yeah, I'm just going to take this away. So I'm going to show you now the other method that I tried. I have to carefully put everything back again. And where is the desk view? Here we go again. Actually, I'll be honest with you. I was actually hoping a little bit to find some of the xylem tissue as well. So maybe I'm going to try from a different place here. Because the xylem it does look quite cool. Let me put the slides away here. I'm going to grab a new one. It's a little bit a question of luck as well. That's a huge, thick sample. And I'm going to carefully press down here. Let's have a look. It's always a little bit, always a risk of actually breaking. So make sure that the surface is hard, so that the slide cannot bend. It's way too thick. You can see all of the banana being squeezed out here. Doesn't matter. We're going to have a look. And where's the scope view? Yeah, of course, I have to refocus, obviously. Some air bubble. Yeah. And yeah, we are basically a little bit lucky here. Let's zoom in. Yep, that's what I wanted to show you. So that's the thing here. Okay, do you see this long thing going, the vertical thing over here? Let's see if I can find a slightly nicer place. Now I'm just gonna explain it, or maybe over here I have to. Yeah, here it's better. Okay, let's go up a little bit with the magnification. Here we go. Shall we add some color? It's do it like this, or is it maybe too dark? I don't know. Yeah. So now I need to add the arrow here. Yeah, and if you see those almost like spiral or ring-shaped structure, right? Here, the round things here. These are all strengthenings or thickenings of the cell wall. And this is a huge, long vessel. It's called xylem, and it's there to transport water in a plant. Yeah, and very often you're able to see those if you take water samples maybe, and so you sometimes also see those spiral-shaped structures. Though it's kind of left over when the plant is decomposing, then those the xylem tissue is very strong and contains those round ring or spiral-shaped structures that kind of make sure that the vessel is actually maintains shape. And then that is something that I bumped it. That can be seen quite easily because of its typical structure. See, and now I bumped it and I can't find it anymore. So this means I have to go down again with the magnification. Yeah, here's another big one. Okay, here we go. I think this one is, good. This one is actually even nicer to see. Let's go up, here we go. Okay, Yeah, here we can see much nicer this spiral-shaped structure. So you can find this actually in quite a lot of plants. I've also done pumpkin, for example. Pumpkin is quite a bit harder. So like boiled pumpkin, yeah, you see those as well. Yeah. So quite interesting. Yeah. Yeah. So these are long tubes. Yeah. So every now and then I'm going to of course pause a little bit and have a look at the how do you say at the chat question. So feel free to write something there. Maybe you want to indicate at Microbehunter or so it's easier for me to find actually your comments or questions. So I want to show you now another way that I've tried. It's kind of a little bit work in progress. Okay, I just want the differences will not be so big now to what we've seen here. But there is something that I want to do, and that is I would like to break open the cell wall of the banana cells. And I have to say I did not manage to do that, okay? So I'm just going to show you the procedure. Yeah, I take a little bit of banana, and you take a beaker. I'm going to put the rest of the banana away. I'm going to eat it later. Yeah. So let's make sure that there's enough space on the table. And here we go. This one also goes over there. And what we do is, we simply add some water. Okay. And we just basically, break it apart. Okay. Now, of course, this will also break apart the cells. And my hope was, and I tried this a few hours ago, again, not successfully. I tried to use detergents like dishwashing powder. I tried alcohol, and I wanted to break open the cells to release the starch grains. Unfortunately, I was not able to do that. So I'm thinking of maybe getting some an enzyme. I have to see if I'm able to get it. It's called cellulase. It's an enzyme that breaks apart cellulose, and I hope that this way I'm able to maybe break open the cell walls. I don't know. Okay. But you get the idea. It doesn't have to be completely homogeneous. Maybe sometimes it's good if there's some chunks left in here. Okay. Peroxide. I've also been thinking about that. Hydrogen peroxide. Okay, H2O2. Why am I not using it? Because I ran out, unfortunately. So this is indeed also a thing that I was considering. Hydrogen peroxide is very caustic. It's very aggressive. It digests organic material. Unfortunately, I realized it didn't have any left. So it's a little bit the pity, right? Otherwise I would have tried it. But of course now, just how do you call it? A strainer, right? This is not a messy part. And then you just basically kind of do it like this. I just leave the rest in here. So you see that all of the larger chunks are kind of removed. So I put this away as well. And you see that there are these things floating around. I was kind of hoping initially that these are the starch grains, but they're not. The things that you see floating around, these are actually individual banana cells. So this method does have a little bit the advantage that the banana cells are not sticking together, but they're more separated. Maybe by adding some enzymes or some other substances, maybe it's able to break them open in the future. I was not able to do that, unfortunately. So I'm going to use this here. And we're just going to grab another microscope slide. Some people have been asking me whether I reuse the microscope slides or whether I throw them away. And I have to tell you, I always reuse them. So what I usually do is I collect them and then I put some boiling water over them. And then I clean them. So, if you wait a minute or so, then you can actually see that some of them start settling on the bottom of the beaker. And then I think I could actually try it. You don't need a lot anyway. So, and let's just have a look, and then let's do a little bit of staining as well, just for the fun of it. This is actually the advantage of this is that the stains will actually reach the cells much more easily. Where is the scope view again? Yeah, let's here they are. You know what? I'm gonna go down with the magnification, right? Are starch cells stored in the cells or somewhere else? Where do they usually get stored in the plant? They basically get stored in the so-called amyloplasts. Amyloplasts are cell organelles that are used for storage of starch. And some parts of a plant are meant to store a lot of starch, like for example the seed, in this case also the banana, certain fruits. In a potato, of course, the potato tuber contains starch. Yeah, and the plant uses, of course, the starch as an energy storage, and of course we humans use the starch as our food. Now, okay, what we see here, of course, looks quite pretty much the same as before, with the difference that the cells are now further a little bit separated. Okay, so this has a little bit the advantage that it's easier to mix in some stains. Do you see those long fibers here? Yeah. No, it does not mean that these fibers were in the banana. It's more likely that they were on my microscope slide as a contaminant. Yeah. So nothing to worry about here. Okay. And on the outside here, I just want to show you again. And one of the things that I found a little bit interesting, it's a little bit of a pity, I was not able to see any other cell organelles in there, like for example a nucleus or something like that. Yeah. But what we have, of course, in here is we have the starch grains, and on the outside here the cell wall, and a very thin cell membrane that we're not able to see because it's so thin. Yeah. Over here, it almost looks like like there is also this internal membrane here. Could maybe be the membrane that surrounds the starch grains, and that's also something that we often see, is that the starch grains are not just randomly scattered in the cell, but they're kind of bundled together in one place. Yeah. Yeah. So yeah. Remember the structural differences between starch, cellulose, glycogen, and chitin. If you are interested in a little bit of biology, starch, cellulose, and glycogen all are made they are in plants and animals. And chitin, that is basically the exoskeleton of arthropods, like insects, for example, and also fungi. Yeah, but yeah. So that's basically how it looks like, and I thought, okay, it's kind of very obvious. What we have to do is when we see starch like this, then of course we have to do a little bit of an iodine staining. Okay, so let's have again a look. So that is the way that it looks like in a regular bright field, and I'm just going to add a little bit of iodine to the whole thing, and you have to be careful when you're messing around with iodine. Because iodine is very, very aggressive. So really make sure that it does not touch any parts of the optics or whatever, right? I also don't want to use any metal tools to get it in contact with the iodine, because it's very corrosive. Okay, so for that reason. Yeah. So where do I have it even? Here it is, iodine, Lugol's solution it's called. Yeah, iodine by it's Lugol. Yeah, I bought this over Amazon. I don't know why they I don't know why they actually sold it. They had some other uses as well. It's not just for staining. Yeah, and the thing that I really want to make clear here is I want to stain it, obviously, but I want to remove then the iodine as well. Okay, you're not seeing that because you need a desk view. So I've taken again a sample just right now. So I'm going to add this one over here. And what I'm going to do next is the following. I'm going to, I need to just save the tip here. And what I want to do now is, if I just add the whole drop here, it's going to be way too much. So what I'm going to do is I'm just going to take a small sample from here, using a clean tip, obviously. And this should be enough. Okay. Just mix it in. Okay, here we go. A whole bunch of bubbles. Okay, cover glass goes on top as always. Sometimes what I do is what you can do as well, and maybe that you get a nicer contrast this way. I don't know if you're able to see this. Yeah, actually, I don't know. You see that there's some. It's slightly, yeah, it's out of focus, but there's some dark spots in there already, right? Yeah. So and what I might want to do is the following: simply to go here on the side and maybe simply to remove some of this here, hoping that I'm not removing yeah, any, yeah, and because I don't want to have too much. This is actually two cover glasses. There's two cover glasses sticking together. Here we go. So yeah, I don't want to have any iodine spilling out on the edges here, right? But you can see now quite well there are these dark dots already. So yeah, the starch grains have been stained, and let's remove this here again. And I am now again changing the camera. Here we go. And here we go. Okay, yeah, that's how it looks like. Yeah, you get the idea. Yeah. So the iodine evidently was able to enter the cells without problems. You see the background is now also a little bit yellowish. Yeah, this is not bright field obviously, and yeah, you can see quite nicely. Yeah, that yeah, has stained them quite well. So what I'm going to do is. Yeah, iodine is used to disinfect wounds. That's correct, but this was not the reason why they sold it. I think they sold it for, I don't know, as a food additive or something. Yeah. So very quickly. Okay, yeah, some communication going on here. Where does the starch usually get stored in a plant? Well, in those parts that are needed for the plant. Basically for food storage. So for example, in seeds, a lot of them in seeds, I mean, when we eat wheat grains and so on, it's all full of starch. I mean, I can show you. Yeah, over here I've got some wheat. You can't, that's, no. Those grains here, now you can see them, right? Yeah, and it's full of starch because when these are seeds and when the plant grows, there are no leaves there yet for photosynthesis. So it's using actually the food storage. And of course, we humans use this also as a food. So that is basically what I kind of wanted to show you almost because there's a second stain that I would like to do, which also looks kind of pretty, but which is not really very revealing in that sense, but it does look kind of nice. And that is staining it with how do you call this? With methylene blue. Okay. And again, this is now the thing where you can experiment. Is this what substances can you use to kind of break open the cells? That's a little bit the thing. Yeah. So we take another sample over here, and where is my methylene blue? Here it is. Yeah. Löffler. He was a German microbiologist back in the 19th century. Yeah, and he has kind of made a recipe using methylene blue, which is ready to use, which also contains, oh, you have not seen this, also contains alcohol and potassium hydroxide. Yeah. So it's used to stain acidic parts. So it will actually stain DNA as well because DNA is acidic, deoxyribonucleic acid, and that's why if you use an alkaline, a basic stain, it will stain acidic parts of a cell. Yeah. So, and let's have a look here again, and then you're going to see that. Yeah, the methylene blue is a general purpose stain. Again, two of them were sticking together. It's a general purpose stain, yeah. And is not quite as, at least the Löffler solution is not quite as aggressive as crystal violet, which I also have, but which is really, really strong. So let's have a look here again. Ah yeah, I have to change to the scope view. Here we go. I need to focus. And yeah, let's go up a little bit with the magnification. So again, that's bright field, okay? And this should already illustrate something here. That's actually the whole point of a stain is that the cells are now more blue than the surrounding. Okay, and that's the whole point. A stain, the chemical reacts with whatever parts are in the cell and actually turns them more blue. There's no point if a stain colors stains everything the same way, because then you're still not able to see a difference. But the point of a stain is that they kind of react with certain parts of a cell, and the rest should remain unstained. And that's actually something that we see over here. Yeah. As well. Yeah. Let me see if I'm able to find something else here. Yeah, here. There. See, they're still clustered together. Yeah. Look, very nicely visible. Let's go down a little bit with the magnification. Nicely visible that the banana cells stain more blue. They kind of absorb the stain, and there is, and the surrounding is white. Yeah. So that's a little bit the thing here. Let me quickly check again lots of comments. Okay, yeah, fine. Punches banana against the desk. I'll beat those darn cells open. I'm just reading. I tried that too. Okay, I tried to actually squeeze the cells apart. I'm going to show that to you. It didn't work, but it ended up crushing the starch grains. Okay, so I just want to show this to you as well. So I've actually used this. Let me show this last try, and then I want to move on a little bit. Okay, I've been already doing this half an hour, and I still have a few things I want to show you. So I'm going to use a very little of this here. Okay, and cover glass goes on top. Yeah, and now I'm going to squeeze this here. That's a little bit difficult now. Why is this difficult? Because the surface is soft, and if I press down, I'm going to break the how do you say the slide. So what I'm going to do is the following. I'm just going to do it the easy way. I'm just going to put this around here, and I'm just going to press it together like this, hoping to break open the cells. Now my fingers hurt. Okay, let's have a look how this looks like. So hoping to have squeezed open the cells, and we'll see. Where is the scope? Now I have to find them. Okay, let me see. Here they are. Yeah, okay. You see, it's a little bit like this, that over here that's still water, that's a hue, that's down here that's an air bubble, and look at this. Now the starch grains, maybe I did manage to squeeze the cells open, but look at the starch grains as well. They are also pressed flat. We can actually go up a little bit. It does look kind of interesting. Yeah, you can see actually how the starch grains are also now fractured and torn on the side. Yeah, let's add a little color. Look. They're still polarizing a little bit. Yeah, because of the polarized light, you see that they're have slightly some colors. Okay, yeah. See that starts to contract a little bit. Yeah. So let's see if maybe there. Let's see if I'm able to find a few other cells. Here are more. Let's see if the cells are still intact or not. Most of them probably. Over here seem to be still. Yeah. You see there, the cell is still intact here, right? But the starch grains are kind of squeezed flat. Yeah. So people want to know which microscope I'm using, and it's an Olympus microscope. It's apparently a very popular microscope which is used in pathology, I was told. Yeah. Thank you very much for the contribution. I've just seen. So I'm just saying the important thing is the following. Many people say, okay, first of all, the colors that you see here and all these things, yes, that's correct. This the optics that I'm using, this does make the microscope a bit expensive. Thank you again for the contribution. Basically, however, I have to also say this, by using color filters, oblique illumination, and so on, you can get a lot. It looks maybe a little more colorful, but you can see pretty much, not more or much less than what you would see in regular bright field, like you've got over here. Okay, And especially if you're using polarizing filters and so on, you can do a lot. Yeah? So you don't need a very, how do you say, expensive microscope. So I do want to comment also to one comment over here, that our saliva contains amylase, which reacts with starch to make glucose. Maltose, actually, it doesn't matter, but that's correct, and it is indeed possible to observe when you add saliva spit to the starch grains that it will start to digest it. I've tried this before. However, the starch grains are generally quite stable, so what you have to do is you have to boil the starch grains first. Then you have to, because it makes them, it makes it softer and more amorphous, and then the enzyme can act better. So it is actually possible to see this also under the microscope. I decided not to do that today because it's a separate video, okay? Because it takes more time. I have to actually heat up the slide and so on. So there is a little bit more, it's a little bit more involved, okay? So this is, yeah. So I'm just reading here, okay. Yeah. So in any case, if you want to see those starch grains, I have the following recommendations. Number one, we've seen bananas, easy. Number two, even nicer looking are potatoes. What you do is you take a potato, you scratch the surface, and you basically you scratch a little bit off from a cut surface. You cut the potato open from the surface, you scratch them off, put that under the microscope, looks beautiful. Number three, possibility number three, you wash some rice, and you take the white rice water and you allow the starch to settle and look at that. Okay, and you can see that the starch grains, they look different. Okay. There's a question. You know what I'm going to do is I would like to answer all of those technical questions maybe a little bit later. Okay. Yeah, just for, yeah. Egg inner membrane. Haha. I didn't forget about that. I did not forget about this. I made a video, and I'll take this as now a, how do you say, an excuse to move on a little bit. I made a video in my other channel, okay, about eggs and how you can make the eggs, store them a little bit longer. And there was a comment, please put the membrane of an egg under the microscope. So I boiled an egg. Okay, so here are the eggs, and I have to tell you, this is gonna be a little bit disappointing for you because the membrane of the inner membrane of an egg is not made of cells. Okay, so we're not gonna see, so it's actually some kind of a structural protein. It doesn't matter, we're gonna put it under the microscope in any case. I have not done that yet. Okay, I actually wanted to, and I said, no, I'm gonna wait. And I'm going to do this together with you. So I'm always very thankful for those recommendations. But indeed, yeah, when I saw the comment to put a little bit this membrane of an egg under the microscope, I said, yeah, that's actually a cool idea. And then I realized, actually, it's probably not going to be super exciting because it's just structural protein. So maybe we're not going to be able to see anything at all. Doesn't matter. We've still learned something. And I also do not know what to expect. Okay, I also don't know what to expect. Yeah, so yeah, so this yeah, the thing is the following, that those membranes, of course, keep the egg white and the yolk and everything together, and also acts as a barrier against bacteria. So what I do next is the following. Of course, cover glass. However, I just realized I need a. Oops, I just realized I need a container. I need to get a, I'm just going to use this here. Okay, here we go. I'm going to use this for my water. And then we're going to have a look. Where is this here? I need a new pipette. I'm going to add a little bit of water here. Yeah, see, it readily absorbs the water, and it's actually a fairly easy specimen because it's so nice and flat. Then let's have a look at it, okay? So again, let's tune down a little bit our expectations, okay? If we see something interesting, very nice, and we're all happy, I do not expect it, okay? Because I'm kind of concerned that it might be very homogeneous. So let me do the scope, but it doesn't matter. Even if it's not super exciting, we've still learned something. Like for example, like we've seen over here, that they're indeed fibers. Didn't expect that. Looks almost like paper. Let's turn it up here. This is actually something which does look more interesting than I thought here. So again, if we just joined in, this is the membrane of an egg, a chicken egg. Okay, so here that's the central part. Let's have a look here. So it looks very much like paper. It's crisscross. Those dark spots here seems to be some air bubbles. But the interesting thing is here on the side. Okay, let's go up with the let's add color. It's almost almost like fungi, like a mold. Let's go up further. So open it a little bit, the condenser. Oh yeah, it's quite interesting. Yeah. So again, this is these are the sides of the skin of an egg. These are then protein fibers. Huh. I'm reading also some comments here. Yeah, there is the comment about DIC. DIC stands for differential interference contrast. And there are a couple of companies that are offering this. You cannot buy those microscopes over online. These are not, yeah. You have to talk to one of the, if you want to buy it new, right? I know if you're interested in this, you can talk to the company Euromax. It's a Dutch company. They're offering also DIC microscopes setups. My microscope is an Olympus microscope, and when you buy those microscopes, you don't just buy them basically finished in a box, but you actually have to tell the company the configuration that you want, and they'll basically buy the parts or put the parts together. Okay, yeah. So yes. Thank you, Jesse. Euromax, they have the Delphi X and the Archeo X. Yes. And you can actually, if you're interested in these, you can have a look at the catalogs online. Yeah. So DIC is, yeah, I have to tell you, yeah, I'll tell you why DIC, I mean, it's always like this. These are not manufactured at large scale, right? I personally think that the price, the high cost of those is not always justified, right? But I suppose the cost is also so high because these are not produced to the large extent as other microscopes are. I mean, you've got polarization. The optics, they have to be a little bit special as well, because otherwise they're again changing the polarization of the light. They have to be so-called stain-free optics. And then there are these prisms in there, which are also kind of expensive. So it kind of adds up a little bit. But actually, from the complexity alone, I mean, it's a polarization setup. It's not super complicated or complex, but the optics have to be right. Otherwise, the optics destroy the polarization again. And in that sense, yeah, I think the prices maybe are even kept up deliberately high. Yeah. Yeah. So BioBlue, yes, the BioBlue, for your info, if you're talking about the BioBlue microscope from Euromax, that's I've also made some reviews on this channel about this. That's the microscope that we're using also in school. Okay, so these are also yeah, decent microscopes, good microscopes for educational use as well. Okay, so yeah, so this is basically the how do you say the egg, the skin of the egg, yeah, and yeah, as you see over here in the center, there's not a lot to see. It's not made of cells, right? But on the side here, we can actually see that the individual fibers, these are protein fibers. Okay, so let's move on. Okay, I just check the time a little bit, 44 minutes. So what I'm going to do now is I want to show you a little bit the more or less success that I've had in growing my little ciliates. If you Yeah, I'm just every now and then checking as well. If you want to buy good microscopes as well, you can actually go into the second-hand market. Okay. So next thing, where is the next thing? Yes, this here and this here. You remember maybe last week? Oh, here we go. Number one. Let me put this here away. The banana goes away. And this here. Okay, I'm going to explain this to you again, what I've done here, for those of you who were not here last time. This is basically a pond water sample. I can show you from the side here. Just, yeah, a little bit of pond water and some stuff. And what I've added here in this larger jar are some cereals, which I'll just show you as well, if I find them. Where did I put them? Yep, some breakfast cereals. Okay, one of them, yeah. If you just take one of those here and have a look at it, that is actually, this one over here is actually a wheat grain, which was also crushed and compressed. Yeah, they're kind of soft a little bit, yeah, and fragile, obviously. Yeah, so oats basically. Yeah, and I've added one or two of these to the water sample. I think this was already 2 weeks ago here, and you can see that it's starting to look a little bit disgusting. You see that there's still some stuff floating around. And then what I have also done is I've taken those grains here. Okay, last week, so last week's live stream, if you remember, I crushed one of those grains between 2 spoons, and then I also dropped it in here. Okay, and what happens now is the following, or what should have happened or should happen is that bacteria now start to feed on that. And then whatever other microorganisms are in there, ciliates and whatever, that they also start to feed on the bacteria, and then they also start to multiply, right? So, so I have done this a couple of years ago already, and I was really happy and massively surprised how many of them were really growing, nice, big paramecia and so on. I'm not quite as successful this time, but still, I'm able to show you that the concentration has increased. This one over here, not quite as much from last week as the one from 2 or 3 weeks ago. Okay, but I'm just still going to show you what I've done, and I'm going to do now the following. I'm going to now. You have to be a little bit careful now, because every time when you're kind of growing microorganisms and bacteria, you want to be a little bit, how do you say, be careful that you don't contaminate, yeah? Especially those tweezers here, right? They're kind of sharp, yeah, so I don't want to hurt myself with those. So look what I'm doing. I'm taking now, let's put everything away here again. So these are the cereals, yeah? Not the cereals, the wheat grains. And I'm taking now one of those here. Okay. I'm dipping it on the slide. It goes back. And this one over here, I'm going to show you what I'm doing now. I'm going to okay, it's alcohol. Okay, I just want to disinfect the whole thing here. Yeah, it's going to be fine. And of course, a cover glass goes on top. There's one over here, lying around here. No cover glass. I just want to have a first look under low magnification without cover glass, because now everything is concentrated in a smaller area. So I see what's there. If I put a cover glass on top, everything's going to spread out, and yeah, the density is not going to be quite as high. So let's put it under the microscope first without any cover glass, just to get a first impression of what is there at all. Yeah, it's pretty active. Okay, so that's how it looks like. Those large circles that you see here, these are all starch grains, of course. Yeah. And next higher. Again, everything is without a cover glass, so I gotta be careful. And again, those round things here, these are the starch grains of the wheat grain. Yeah. I was unfortunately not able to identify those larger ciliates here because they do not look like, how do you say, like the classical traditional paramecia. But what I'm going to do now is I am going to add the cover glass. Okay, so something is here. And then I'm going to have another look at some of your comments. Yeah, some form of a hypotry could be, but I tried to find it even in my book, the identification book, but I could not find it. The biodiversity is simply too large. So you now see everything spread apart much more. Okay. And wow, look, quite a bit of stuff going on here. Okay. Yeah. So the starch grains, they themselves also prevent the cover glass from kind of sandwiching the ciliates. Yeah, so they still have a lot of space to move, and that's how it looks like. Yeah. So usually there is some kind of a progression as well. With these starches, do you use polarizers? Yeah, I'm gonna use DIC right now. So this is now polarized light. Okay. So I'm able, So plenty of activity here. So I will tell you another thing that I tried and that I was not successful. Okay, I'm just showing you so that you know what I've been trying to do. And this is the following. But don't worry. Maybe I'm able to manage this somehow in the future. Look, very briefly, these are copper coins. And I added some water, and they're pretty corroded already, oxidized already. And I added a little bit of water to those, to this sample over here. And I was kind of hoping that this would inhibit them somehow because copper actually is disinfecting. Many microorganisms don't like copper. And unfortunately, I was not able to see an effect. So maybe the concentration of copper was not high enough yet. But I just wanted to tell you a little bit of the experiments that I'm doing. So adding a little bit of this copper liquid. Yeah. So that's basically another thing that we see over here quite nicely. Maybe we have to wait a little bit longer. Is that over time, those microorganisms, you see it over here a little bit. Where is the arrow? Huh? Look, it's almost like here that they were spread around and now they're gathered over here and they might start moving now somehow to the side. Okay, you can see that happen quite often because what happens is that the oxygen starts to run out here and then they start to move more and more towards the edge of the cover glass where the oxygen concentration is higher. So after a couple of minutes, we probably would expect more of them to kind of accumulate on one side of the, sometimes they also gather around air bubbles, that we have over here. This takes a little bit of patience, but it is possible to see them actually follow certain gradients as well. Look at, this one is actually quite nice because this one is kind of squeezed. So we can actually have a better look here. Yeah, this is a typical example of one that's in pretty much distress. Where is this? Let's turn this up, Look, there seems to be some kind of a biofilm here as well. It might be all bacteria. Yeah. And if you look carefully, then you can see that in the front and the back part, there are the cilia, the hair. They have bundles of hairs in the front and also in the back part here. Yeah. So. And it almost looks like that all of the rest over here, this seems to be possibly some kind of a biofilm of bacteria. So, so maybe I missed it. How did you get the whole culture with this oat? It was quite simple. I show you. I took a water sample. you know what I could do? I don't have another beaker now here with me, but for those of you who have not seen this yet, let me see if I can get an empty beaker somewhere. Here, a jar. Look, if you want, I can show you what I've done. Okay, and yeah, and this is how I go back to the desk. Maybe this is a little bit too big. It doesn't. Yeah, that's a bad one because it's difficult to go in, right? But just to show you of what I've done is I have taken a water sample. You know what we could try? We could try this pond water sample over here. I don't know if there's so many in here. No, I'm not gonna use this because I see that the water fleas, Cyclops, maybe Daphnia in here. Not a good thing because they eat those ciliates. So that means if I've got even one of them in the sample, they're going to eat up all of them. I've once had this already. I tried to, I was kind of wondering why I was not able to grow them. And then I had one of those guys in there and it was eating up all of them. So I'm going to take the other water sample. So this is the one. Yeah. It looks disgusting here on the top. But actually, there are lots of these cells floating around here. And yeah, so what you do is the following. You just take some of this here from the surface. Most of them actually accumulate on the surface because this is where the oxygen is. Okay. I think, and then I have also another one for next week. Oh yeah, thank you. The questions are here again. I think this is again dirty, so I don't know where to put it. I'm gonna put it over there. And I put this away again. Okay, and then I'm gonna have a look at the others here, okay? And then you need one of those grains here. And what do is you kind of you crush them. Here's one, right? I don't have a spoon with me now. No, there is one over. I'm gonna just use the. Yeah, I'm just gonna press. I'm just going to press it on my table, okay? So just in case you're wondering what I'm doing. Okay. I've kind of crushed it now. Here it is, yeah, so the starch is exposed. I drop it in here and I leave it in there for a week and I hope for the best. Okay? And this way we should get a lot of them. Now, the problem with those cereals, with the breakfast oats, is that they kind of are too fragile. It's too difficult to take them out. So it's a practical thing. So, but I would like to show you now, or let's have a look at this one over here. Okay, I'm going to use now a pipette to extract some of them. So I go into there where the cereals are, and we do the same thing. Cover glass. Okay. I always have to check that, yeah. And we have a look here. So this one is a little bit older. Okay. And let's have a look now here. Oh yeah, I have to change again to the microscope view. Let's... Not very... let's see. Check here. Okay. Ah, yeah, they look already a little bit bigger. Or maybe not, I don't know. Yeah, so basically it depends a little bit on luck, on which ones you actually, which ones actually multiply. Yeah. There are more of them, more different ones over here. But they also seem to be kind of similar, having those. Yeah, and then you go up with a magnification, and then you hope again to be able to see something. Yeah. If they are moving around too quickly, you have a couple of possibilities. Possibility number one is you simply remove water so that they're kind of sandwiched, just like we've seen before. Or you add a substance called methyl cellulose, which is very viscous and which also limits their movement a little bit. Okay. Okay. Anthony has just mentioned, okay, that you've got a Euromex Delphi X. Some people have been asking a little bit about my camera setup. I'm using a DSLR camera, and basically without any intermediate optics. So the image that you see over here directly comes from the objective. So any intermediate optics might lower the image quality, but this one does not have any intermediate optics. So its design was just designed in such a way that it works without that. And the interesting thing is the following. Normally when you take a picture using a camera, the field of view is smaller. But in my case, with a camera, I can actually see more than by looking through the eyepieces. It's kind of interesting. Yeah. Okay. So this is a little bit, just something I wanted to share with you. So you see, it's a little bit work in progress. I hope that I'm able to get a yet higher concentration, because a couple of months ago, as I said, I had, I don't know, several thousands of them swimming. Yeah. This one over here is dividing here. Okay. Last week, maybe you remember that we've also seen a dividing one. Yeah. So that's the question. Okay. Where does the blue light come from? Look, I can change the color. This is because of the prism that I'm using, right? So this is not because of a filter. That's because of DIC, right? So that is basically this technique, and I can play around with the colors and use whatever I want. Yeah. So that's because of the prism. Yeah. And if I want to basically have it completely natural colors, I just switch it off. You see how much brighter it is, because this technique really uses a lot of light. And this is now regular bright field, and these are now the natural colors. Okay. Yes, there was a dividing cell up here again. Yeah. I think the one that we've looked at last week was actually a little bit bigger. Yeah. Yeah. So you get that. Yeah. And all of, again, tons of, you see all of those tiny little things that you see swimming around in the background here is all bacteria. Yeah. Yeah. Okay. Just wanted to I've been with one eye. I've been going through the comments a little bit. I'm just now checking the ones in red, which are basically questions for me. Yes. Okay. Exactly. This was the egg membrane comment. Yeah, I'm just scrolling up right now. Send me off topic if you do request a letter. Egg inner membrane. Yeah, I've done that. Thank you again for the financial contribution. This goes all, just for your information, all of this money goes on a separate bank account, and then I buy microscopy stuff for that, slides and so on. So basically I kind of show it to you, the things that I'm buying with that. The question about the starch cells. Peroxide, I ran out of it. Dual plants. Okay, here, by PKN, posted at 9:47, so it's already a little bit earlier in the chat. You know what? I might be actually looking at the how do you say? I'll be looking at the questions here, and while I'm going through the questions, we can have a look at the all of those. Let me put a nice. Usually I like a blue color because it kind of imitates water a little bit. Okay, so let's do this here. The video. So I've got the question. Some of the questions and comments here. The video where you ate the brown apple was so gross. I love to see it. Yeah, if I made a video on my other channel some time ago where about oxidation yeah. So when apples turn brown, why do they turn brown? And I've frozen an apple, and yeah, it tasted like a normal apple, but it turned all brown and it was very soggy. Right. Actually, thinking about it, I could have put that also under the microscope. Do plants rely on capillary action to transport water through the vascular system, or is there some active pumping involved? Both. Both. Basically, in the xylem, when water moves up, this is a passive process by capillary action. However, there is also a little bit of active process involved because the xylem is loaded with ions. So basically, there are membrane pumps that pump into the xylem some ions, minerals, and then water follows by osmosis. The water itself moves up on its own, so to say, but it does require a little bit of energy to get simply the ions into the xylem. So it's actually a combination of both. Yeah. Starch cells stored in the cells or somewhere else. Where do they usually get stored in the plant? Well, in a plant, let's put it this way. How shall I put this? All of the starchy parts of a plant, like banana is mostly starch. It's why it gives us so much energy. Potatoes are starch. All of the seeds, beans, for example, wheat grains, like we've just seen, all contain starch, right? And because the starch is an energy storage for the plant. Aniline blue is the perfect stain for this. Interesting. Didn't know that either. What does our stomach use to get the starch out? Starch digestion happens both in the mouth and also in the small intestine. There's the enzyme, I think it was already mentioned anyway already. There's an enzyme called amylase. And this amylase breaks down the starch into maltose. So it does not break it down all the way to glucose, but into maltose, which are two glucose molecules together. This is absorbed into the blood, and then the cells, our body cells, they will then break down the maltose into glucose. However, if you eat, for example, raw potatoes, very difficult to digest because the starch grains are very hard and crystalline, so cannot be digested easily. So you have to boil it, and then the starch becomes amorphous, and this basically means it soaks up the water, becomes much softer, and then you can also digest it better. A little bit of biology. I'm a biology teacher. Yeah. Interesting. Surprising, you could crush the grains and get and not break the cells. Yeah, this is also kind of interesting. Maybe this shows a little bit how flexible the cells are. Remember when I crushed the banana cells, right? This is also something that I've seen. Look, right now, sometimes the cells, they stop moving, and then all of a sudden, when one of them bumps into the other one, all of them start moving at the same time again. I'm using Olympus microscope. Okay, a Novex BB8. So the comment by Sami, I have a Novex BB86. I don't know that. Which one? Yeah. I could research this here. Huh? Egg inner membrane. I've done that. Okay, then the oats I've done. With these starches, you could use polarizers to see them better. Yes, you can do that. Yeah, that's also what I have done. What I've also done some time ago, it's kind of interesting. I still have got some of these Iolozola worms in one of my jars. And if you feed them the starch grains, you can actually see how the starch grain travels through their digestive system. Also pretty fun to observe. So let's have a look here. Sometimes people ask me, well, do you know which ones, what they are, what these cells are? I say, well, identifying them all the way down to the species level is, for me, practically impossible. I mean, you can maybe determine a little bit the direction, but in order to really know what they are, microscopy rarely is sufficient. You probably have to do some kind of DNA analysis or whatever. Over here, those round circles, I wonder what they are. If there's some kind of, I don't know, some cysts or something. Yeah. But as the time goes by, you see that those cells, they start to become wider and flatter. And the reason is because they're compressed between cover glass and microscope slide. Yeah. So let's go here again. Yeah. But they're still able to move around fairly quickly. Sometimes when they don't move, you can actually really see what's going on inside the cell. And that's actually then quite interesting to observe as well. You can actually see them also take up food and make food vacuoles and all these things. Yeah. So yeah, but why not? Yeah, I not only have you see it's pretty compressed already. I not only have DIC, some kind of round cysts. I don't know what they are, but I also have yeah, one phase contrast objective. Okay, and yeah, one. So I'm going to show that to you as well. I've got a 40 times phase contrast. So I need to switch this over to here, open it, and phase contrast looks like this. No, I need to take the filter out to make it a little bit brighter. Yeah. And phase contrast actually is better to be used to observe bacteria because they appear to be darker on a brighter background. Yeah. So yeah, right now you can also see the cilia, those tiny here, yeah, here in the back better, yeah, and also here in front. Yeah. So I'm going to quickly check if there are any more comments here. For ID to Generosity Feusner, yes. I do have a book. Look, I just show you the cover. Just a second. But unfortunately, I'm a little bit afraid to actually open it and show it to you for copyright reasons. But I think the cover I can show you. That is actually my favorite book. It's in German, but it does not matter because it's actually full of drawings. Okay, then there is once you have got the drawing, then you have got the name, and then you can type in the name into Google or whatever, and you can get more information about it. And the description of the different organisms in there, yeah, basically where they appear or where they can be found and so on. But the real important thing is actually the drawings. Okay. Yeah, so that's the thing. Question is, can you do real sperm? Then I would like to refer you back to, I think was it last week or 2 weeks ago. I was somebody sent me really nice videos of spermatozoa. 2 weeks ago, and really probably the best ones that you can see. Yeah, so spermatozoa. I've 2 weeks ago made a live stream. You can actually check it. All of the videos are online. Yeah. If you can read German, you do not even have to be able to read German, because what I recommend is that you take yourself a translation app. You take a picture of the paragraph which describes the organism, and then you can get it automatically translated. And I would say the descriptions are not really so super important. I'm just going to, basically, it says something about the size, which you don't even, where it can be found. Yeah, and some general info. I'm a little bit worried about actually showing it to you for copyright reasons again, but it is a very very good book, and even if you get an older edition, I recommend it. The question is why are these small ciliates are dying so fast? I saw the same in my observations as well. Well, those ciliates over here, the small ones, I don't think that they are dying. They're just resting, and then as soon as somebody bumps into them, then they start moving again. Okay, yeah, you see. So, which microscope camera software do you use? I'll be totally disappointing you. I'm not using almost any. What I'm doing right now is I've got the camera connected into the computer, and I'm using a program called OBS Studio, which is a free program where I'm able to do also live streams. Yeah, so OBS Studio is the software that I'm using. However, theoretically, I don't need any special camera software. I can actually use the one that's built in Windows to actually look at the things. Yeah, so yeah, that's the thing. You see, there's a one over here again dividing. Yeah, and those tiny dots that you see again, these are bacteria. Yeah, here in the background. Yeah. So let's go back again to, yeah, that's a little bit, that's again, yeah, DIC. Yeah. You can play around a little bit with the colors. Yeah. Yeah, my suggestion, my suggestion is don't let the colors and the equipment and the technology kind of deter you. If you say, oh my God, it's so expensive, all this stuff, you don't need it. You can do a lot of interesting microscopy stuff also with microscopes that cost only a few hundred. Dollars or euros. Yeah, I'm serious. Don't let this deter you. And there are plenty of opportunities like using oblique illumination and I don't know, Reinberg illumination and polarization. You can, do lots of crazy things here. This little long worm-like thing that you see over here is not a worm, obviously, but that is most likely some kind of a cyanobacterium. Yeah. So yeah. Yes, exactly. Yeah. If you have just for your info, it's certain dedicated microscope cameras do require specific software, or you have to install drivers that you're also able to use it without the software. So that's direct draw or direct view, I think it's called, or direct show. There is this. Is it direct draw or direct view, direct show, something with direct, and that's a driver that allows you to use also a regular camera, a microscope camera, also to be used in other software. Okay. So, yes. Yeah. So it's kind of fun to observe them. Sometimes that's what I like doing. I just like to watch them. Yeah. Yeah. Again, there's a dividing one over here. Yeah. You get the idea. Okay. Then I see. I'm reading the comments again. Yeah. What's the length of the bacteria filamentous form? Well, let's put it this way. I don't have a micrometer slide right now, but generally bacteria are approximately in a range of 1 micrometer across. Yeah. So yeah. So let's have a look again. What time is it? I still have maybe a couple of minutes. Okay. Now, if you maybe I'll explain this. If you kind of wonder what in the world's going on here, why is there this dark spot in the center? And that is because of the phase contrast filter. Yeah, so I have to change over. Yeah. This actually looks quite nice. Yeah. Also, a couple of, how do you say, if you're interested in cameras and so on, a couple of months ago, if you check again in one of the live streams, I presented some mobile phone adapters and also some ways to connect the camera to a microscope. Yeah. So plenty of, especially in this channel, I've made already plenty of videos demonstrating some adapters. I mean, honestly, there is no system that is absolutely perfect. Each system has its own advantages and disadvantages. Some are, very cheap, but it's not very convenient. Others are quite, elaborate, very bulky as well. There is actually no I would say no camera setup that is perfectly ideal. And each one is some kind of a compromise of what you want to do. If you want to make, good point. Let me talk about this. If you want to make videos for YouTube, do you recommend that buy a €150 camera or use the phone adapter? Okay. The thing is the following. If you want to make videos on YouTube, it doesn't matter so much as long as your setup is reasonably stable. Okay. Very often I see very shaky videos and so on. So my recommendation is make sure that it's reasonably stable. With a mobile phone, you can get quite decent results. However, when you take a mobile phone and take it through the eyepiece, you're going to see the image in how do you say in a circle, right? So you probably want to zoom in afterwards. right? And so it's more of a convenience thing. If you buy yourself a €150 camera and you connect it over USB, be aware that USB is sometimes slow. That means you do not get the frame rate. Yeah, so you have to reduce the resolution. And then you can basically hope that the frame rate is high enough. So especially a mobile phone has the advantage that you get basically the full frame rate. But if you use a USB camera, it might be too slow unless you go down with the resolution as well. Okay. Okay, I can say it's an Olympus BX53. That's my model scope that I'm using. It's configurable, so the cost depends on the objectives, on the eyepieces, and whatever you want to have. Okay, and I'm not telling you how much you paid for it. Okay, so that's yeah. So the how do you say, those microscopes are always sold with never out of the box, but you always have to get yourself an offer from the company, and then you have to specify exactly what you want, because they will buy the parts, or not buy the parts, they will put the parts together. And when my microscope came, it did not come pre-assembled, but it came in, I don't know, 30, 40 different smaller boxes. Everything was packaged separately. I had to put it together myself. Well, actually, I didn't have to. They would have sent someone from the company to put it together for me. That would have been included in the price. Yeah, but I chose to do it myself. Yeah. So what is the advice? Reducing skipping? Do you reduce the resolution or the recording, or other suggestions? What you have to do is the following. Skipping is you have to go up with the light intensity to reduce the shutter speed, to make the shutter speeds short, and you have to go down with the resolution. That's really important, because normally when you are taking very often when you're using a microscope camera, then it's set up to the maximum resolution, which is way more than a video resolution. Okay, so skipping and these are not a problem when you use a mobile phone, right? But if you use a microscope camera, then there is a problem that sometimes it goes irregular, and that the reason why that is because you, without knowing it, maybe you use the maximum resolution, which was way more than what you normally use for video. So you have to go down with that. Yeah. So that is a little bit the thing here. Do I have dark field? The answer is semi. I will show you what I mean. Very good point. I have phase contrast, and phase contrast uses a phase contrast annulus filter, which can also be used as a dark field filter. So I'm going to show you, this is now my 4 times, and now I use phase contrast, okay, and I have dark field. So I'm kind of abusing my phase contrast filter for dark field, and this only works for the 4 times objective. Okay, so this is a little bit the limitation that I have. Yeah. Okay, so yeah, I found a 1080p camera at 100 frames per second. That's pretty decent. Okay, that's pretty decent. Yeah. Basically, 30 frames per second is the one that I'm working with. So, for example, my camera that you've got here is able. Right now, I've got 1080p, just regular. It's HDMI, and the HDMI is converted to USB signal and goes into the computer. Theoretically, I could record with my camera. I could record 4K into the camera. I'm not doing that because I don't need that, but theoretically I could do that, but I'm not able to send 4K over the USB cable. It simply is too much, and also if I send 4K over HDMI, it needs to be converted. It simply requires way too much resources. Yeah. Okay. So most of them, 30 frames per second is fine. Yeah, just want to show you something. Just because we were talking about how do you say that's the wrong one. We were talking about how do you call this frame rate. Okay, let's have a look. What I'm doing right now is the following. I can actually change the shutter speed of the camera. Look what I'm doing right now. I'm turning a wheel, and I'm going to do now the following. I'm going up with the shutter, so exposure time, and I'm turning down the light again. Okay? I'm turning down the light, and I'm going to increase now the exposure time of the camera. What do you notice? Okay? Yeah, do you see actually the motion blur that we have now? Right? And that is now not because of a transmission problem over HDMI or USB, but simply because I have reduced the shutter speed so much, right? Yeah. And then you get this motion blur, and I compensate this by going down with the light intensity. Yeah. Yeah. So this is basically something that you would see if the speed were too slow, right? So, and I'm going to go back again. So what I'm going to do is I'm going to turn so I have to make this dark. So I'm going to make the camera. I'm going to increase the shutter speed with the camera, and I go up with the light, and now I have a smooth movement again. Okay. So I'm lost. I watched all your videos about cameras. I really don't know what to buy with my tight budget of 150. I will tell you what you should do then. Then buy yourself a cheap mobile phone adapter and explore all of the possibilities with the mobile phone first. Okay? So that's definitely my recommendation. If you buy a mobile phone adapter, which should not cost, I don't know, I bought one for €7. Exploit the possibilities of the mobile phone first. You can also connect the mobile phone over USB directly to the computer, like a webcam. That is possible. You need to install an app in the mobile phone, and then the mobile phone can be used like a webcam. You can have a direct USB feed into the computer. Or if you don't want to do that, you can use also Wi-Fi to actually feed the video over Wi-Fi into the computer, right? So my definite recommendations is don't worry about the budget right now. Start really with the cheapest possibility, and then work yourself upwards, because then you are able to see all of the advantages and disadvantages, okay, of a certain system. Yeah, that's generally what I recommend. Yeah, it's always. Try all of the cheap options first, and then you're gonna see what works, what does not work, and then basically you have then a better basis to make a decision. Okay, I've bought a mobile phone. Honestly, there are really fancy mobile phone adapters out there. Honestly, that's kind of funny. The best one is the cheapest one I got. I got one from AliExpress for €7, and it was really stable. Right, so yeah, it's really interesting. Yeah, but ultimately, much is a question of convenience as well. And what I recommend when you do, and mobile phones does have the advantage that you basically get a very smooth image and without any delay over the USB. Yeah, so that's yeah. In that sense, it's convenient, but for doing a lot of pictures, obviously, yeah. What you can, if you have a photo tube, then of course you can also connect the mobile phone on photo tube. What I've done some years ago is I actually got myself a spare camera, a mobile phone camera. I got myself a spare mobile phone and simply had it connected to the photo tube of the microscope all the time. Yeah? But last question, you have two types. One goes in the eyepiece and the other one hovers above. Basically, about what? The one that goes into the eyepiece, that is not a mobile phone camera. The one that goes into the eyepiece is a regular eyepiece microscope camera, which is connected over USB. Okay, that is the, so it's an eyepiece camera. And the other one way that you can do is you keep the eyepiece in the microscope and you connect a mobile phone over the eyepiece. Okay, So that is yeah, the possibility. You can, yeah, sure, you can send the links. I don't know if you can try that, or you can send me an email or so. Okay. So, but my suggestion is if you watch some of the videos that I made before, I think that should clarify a lot. Yeah. So yeah. So the one that goes into the eyepiece instead of an eyepiece, these are the eyepiece cameras. Look at it, it's not fun whizzing around. Okay, I gotta go philosophical again before I stop. Look, I assume that that those all of those ciliates here are basically the offspring of, let's say, one cell. Which, so maybe they're genetically the same. And for me, what fascinates me is, sure, but this whole concept of what it means to be an individual then is really fascinating. So there are no parents, there are no children, you know? Yeah, so, and that's immortality, yes. Yeah, so essentially a cell, they live forever because they kind of continue to live in the next generation, so to say. Yeah, so in that sense, they do not have a built-in lifespan. That's the fascinating thing, yeah? Yeah, because they simply divide and then it goes. Of course, they can die when it dries out or when they're eaten or whatever, right? Of course, this can all happen. But they do not have a built-in lifespan. And that's the fascinating things, yeah? If no parents and no children, why do we call it two daughter cells? Exactly. Yeah, who came? Side issue. Sometimes I'm wondering a little bit. It would be interesting to go a little bit into the history of science and figure out who came up with all of those names. Yeah, daughter cells, yeah, and so on. Yeah, I think links don't work in YouTube live chats. Ah, okay, that could be. Yeah, copy pasting maybe. Yeah, this is yeah. I'm reading this. Yeah. By the way, also USB 3.0. Yeah, your computer, of course, most of computers these days are able to handle USB 3.0, of course. So your computer must be also fast enough. Yeah. Yeah, folks. I think. Look at this one over here. I think I'm going to call it a day again. Look, this one is trying to squeeze itself through here. It's getting very tight between the microscope slide and the cover glass. Yeah. Yeah. So, and I think I'm just gonna call it a day today. Again, 1.5 hours. Look, I'm the thing that stresses me out every week a little bit is I have to think of somewhat topics to talk about and to show you. That's a little bit the thing. So if you have any recommendations or wishes, I'm always very grateful for that. Sometimes I'm just experimenting and trying out things without knowing the result myself, right? And yeah. So, and if you want to, if you, I don't know, have any specific wishes or so, if you tell me a little bit in advance, I will try to, yeah, to do that. By the way, yeah, actually, I just also wanted to show you this water sample over here, which is already over 3 years old. Maybe next week. Okay. Yeah, I want to yeah, say then all the best. Yeah. Check out the videos that I upload every now and then. Also on the other channel where I've been talking a little bit about food biology and so on. If you're interested. All the best. Happy microbe hunting, and hope to see you again in the next week. Bye bye.